Let's talk about P10. P10 is a phosphatase that dephosphorylates the PIP3 right here to a PIP2. Without it, PIP3 remains and we will still get continued ACT and PDK activation and subsequent bad inactivation as we see here, right, by the 1433. If we look at what BAD does in the previous figure here, we see that BAD's job is to inactivate the BCL2 protein, which inactivates the BH123 proteins from aggregating on the mitochondrial surface to release the cytochrome C. This means that we're going to be in this state constantly. It's the BCL2 is always going to be on, and the BH123 proteins are not going to aggregate, and we're not going to get that release of cytochrome C. Without that release, there's no aptasome creation seen here, and there's not going to be any caspase cascade that's going to form, meaning that we're never going to have cell death. And we're going to have continuous cell growth. And this is why we see in cells before the P10 mutation that they are dying due to the radiation, because those cells are still able to take in the P10, the P53's apoptosis signal and use that to create these aptasomes with the caspases in them and create the caspase cascade. Whereas in the mutated model, we no longer have that around, so we're never going to go down this route, and we're going to stay within the BCL2 hindering the cytochrome C release from the mitochondria. Thank you for the class, and that concludes my video.